Hello, my name is Dennis McCormick, and I'm the Director for Genomic Services and Technology Assessment here at the Ontario Genomics Institute, but also for the Center for Applied Genomics at the Hospital for Sick Children here in Toronto. Now, you may not be familiar what a Director for Genomic Services may do, but what I try to do is work with multidisciplinary teams of scientists to bring technologies involved with DNA, proteins, metabolites, and technologies to help determine levels of those, sequences of those, variants that may be associated with disease. I bring these to these research groups to help speed up discovery in their own research projects. Now, what we find are people that are sort of on the bench tend to be a little bit further away from technologies as they develop. So I really try to keep current with what these current technologies are and how I can better bring this to the scientists that are doing the cell biology or the everyday bench work. Now, how did I get into this type of research or into this job? I'm actually not really sure if I looked for it on my way, but it just has happened that my career has sort of evolved to come here. I was trained in molecular biology as an undergrad. I moved into plant genetics as a PhD student. I went into molecular biology and genetics as a postdoc. I did physics. What I was always fascinated with was the ability to be able to get into labs that helped me answer a specific type of question. So how I got into this line of work has pretty much happened through the course of time and being trained in a bunch of different fields. I was recruited back to Canada when they could not find bioinformatics type people. Now, bioinformatics is the study of information, and when we look at data, we're generating so many data points now from huge studies on disease. And really, when you think about data, it's more like oil of the 21st century, really, because what we're trying to do is mine the information in there to sort of associate that with different parameters. As to the next big question in science, I'm not sure that there are many big questions, but I think one of the questions that I'm always going to be a little bit concerned with is how the public perceives science. And sometimes people perceive science to be scary when they don't really understand the basic technologies that may be involved or what we're doing. So I think what I'm involved with, not only during my day job, but a lot more in my free time, is how do I educate the general population that science isn't really scary. And as if you can see in the back of my office, I'm really engaged in the convergence of art and science. I tend to work with artists or do a lot of my own stuff and build installations that help us probe what's going on in science. And actually, one of the artists, well, a group of 4 artists that I'm working with, James Fowler here in Toronto, is I have given him my PAX3 gene. Now, PAX3 gene is involved in phase formation, and one of the genes. There are a lot of genes involved in phase formation, but this is one. And so what I did is I gave him the sequence and told him that he should interpret how my face was going to look. And anyway, so when you look at this painting, it took him a long time to do this, but when you look at this painting, you see from a distance that it's my face, but as you get up closer, it's my DNA sequence. And now I'm working with 3 other artists to sort of take the same gene and interpret my face. And what you'll find is that each artist has a very different interpretation of what that could be.